Is that okay?
Yes. Yeah, you got it perfect.
All right, folks. Good morning. So I'm Zan Luthi-Schulten. And so first I want to say, Klaus really wanted to be here for the first part on the NAMD VMD, but we both came down with cases of pneumonia. Mine got cured pretty quickly. His did not. And he's just not feeling well enough to travel. So we asked Juan to come, and I think he is the one who came up with the structure of this virus HIV capsid. So he knows large systems extremely well. And hopefully through his talk and through Jody's talk, you get more of a feel of, NAMD has been primarily geared for those million and above systems. So as I said, I don't know how many of you are working on such large systems, but if you do, then I can, without any reservation, say you should be using NAMD. We like NAMD for a whole bunch of other reasons, and it is competitive even with other programs for smaller systems. But if it's really small, then I would tell you, do try, take a look at Amber, take a look at GROMACS, and to work on those systems, or work your way up if you want. Okay, so in today's lecture, I'm going to try to transition a bit from the molecular to the cellular world. So I'm going to be cutting a little bit short these first parts, because originally we were giving this talk on evolutionary concepts and multiple sequence analysis because I felt there's a tendency when people do MD, you look at one structure and one sequence, and you fall in love with some interaction, and then you really should take a look if that set of residues is in there, in that protein, in all the other organisms that are out there. And so we're very fortunate. We had at the University of Illinois the sort of this generation's Darwin. His name is Carl Vose. He's the one that came up with the third domain of life, the arches, and established the three domains of life, which got him the Crayford Nobel Prize. So he liked VMD so much, he sat down with us and said, these are the features I really like into a sequence analysis tools. Can you put that into there? And we did, and he was so happy with it, he called VMD Multiseq a tool to think. And that's a direct quote from him, that it is just far from a simple visualization tool for biologists. It's a true thinking tool. And without it, a whole class of biological hypotheses simply would not exist. So when you use the sequence analysis, it is really set up to do evolutionary comparisons. But as I was telling someone before, with the growing field of synthetic biology, there's a need also for bioinformatics. It doesn't have to necessarily be on the comparison level between bacteria, archaea, and eukarya, and those tools can be used anywhere. But if you're working on a bacteria, if you're working on an archaea, or if you're working on something in the eukarya like humans or yeast, then I encourage you to check the sequences from the other domains of life. But the tools can be universally used, so that a sequence alignment on the side here is all from the so-called methanogens, and in interpreting the genetic code, you need these proteins called synthetases, and these guys were missing the one for cysteine. So it turns out it's like a two-step mechanism. It actually recognizes O-phosphoserine, and it has its own little special synthetase that does this. This is something that Carl was looking for when he published the first genome, and that was of the Yanashai that was in this branch of the archaea. Okay, so as I said, when possible, we use always as the reference point the 16S rRNA in comparing organisms. That's what Carl used. And we'll be extremely interested in anything to do with the ribosome. So the talk is more or less over all the molecules involved in the translational pathway in cells, but you can do it with any cells that you want. It's just we have the most experience there, and we can show you what sort of questions you can ask and how you can go about answering them. Okay, so first of all, I think, as I said, I think most people realize now they really should be looking at more than one sequence. So at the very least, you've probably used some sequence editor. These are ones that were taken from one of the databases, and you can see there's a number of organisms here, and it's color-coded by some similarity score. And there's so many different similarity matrices, you really have to get in and pay a little bit of attention which one you want to use, whether you use a PAM, 20, 40, 70. All that means it's been tested and developed, the interaction percentages on sequences that were either 20% similar, 40% similar, or 60% similar. If you do this and compare the sequences and try to do the alignments, you'll pick up important sites, like active sites. Maybe there's something for protein-protein interaction. Chances are that's going to be conserved. Or there'll be communication within the protein. Those will most also likely be picked up through a conservation analysis. Then there's always a question of how many sequences to use and where do you find these. I always just put this in because when I first taught this at the University of Illinois, we're going out and grabbing them from a bunch of databases, many of them at NCBI. One student asked me, he says, "Is this legal?" Right? And I go, yeah, those are our national databases. You can take anything you want from them, right? So it is a question how many, because the databases still are really biased towards bacterial systems. You have tons of sequences from the bacterial branch. It's, you know, it was so hard to do the sequencing on the eukaryotic systems. There are just many fewer. And archaeas were practically none until Carl got the whole field going. So that bias is still there. If you're doing a comparison, then it would be good. Not only with the sequence name, you put the grouping. Are those from eukaryotes or from the bacteria? Now, where this really came into being important was for about 10 years was this huge activity to do protein folding. And then now I would say we're getting into the era of looking at RNA as well. And so if you're looking at folding or structure or function, again, you will want to be looking and examining, if you can, molecules across all the domains of life, and you can get insight into how they fold and what residues are important. And one of the things I did way back when this field first started was to come up with this energy landscape picture of it. This is our famous funnel picture that I did with Peter Walnes and Jose Onachek. But you can also look at conservation where these two subunits interact with one another. So there are lots of applications. So when you go into VMD and you go into those extensions and you click on multi-seq, this is what you would see. So again, we've read in a lot of those synthetases. I'll have a blow-up of this. Up here, I have the structure. So these are synthetases that read the tRNA. They can dock to it, and then they charge the tRNA with the amino acid. So it's these proteins that set the genetic code in every organism. So if you look at the structure of the synthetases, there doesn't seem to be much variation, and there aren't among the class 1s. But if you look at the sequences, there's a lot of variation, but a lot of conservation, say, just in this position here. And if you want, with VMD, multi-seq, you can map that sequence conservation onto the structure. Instead, I mapped the structure conservation onto the structure, right? All right, so when you use that, and to use it to its full capabilities, you also have to bring in some metadata information. Now, we're going to concentrate today mostly on the cell simulations. If you want to do the full exercises, we have all that information in a Dropbox that we prepared for a presentation at Berkeley. We can direct you towards it so you can do all the tutorials. But essentially you need, and I say metadata information, and that includes like what domain of life that is, what's its accession number, what's its uniprot number, and then you have to have some algorithms to perform the alignments, and then another algorithm to perform the phylogenetic tree analysis. To avoid taking, say, too many, say from the bacteria, we have an algorithm called QR, which takes out and saves for you the ones that are most different within any one domain of life. Okay? If you want to search the databases, you can use BLAST, SciBLAST, any of the typical search engines, bring back the results. The alignments are never good, so we have built in not just a sequence viewer, but a sequence editor. So you can remove the gaps, move things together, throw out whole sequences. And then there's a whole bunch of tutorials that are designed for it. That's how it first started in 2006. And since then, we've added a lot of other tools to this. So if you're going to look at the whole genome, you're going to want to use the signatures and the zoom feature. Do this on a bigger machine. You can bring, if you want, the whole Swiss prot database. You can bring in all the RNAs. It's in a database called Green Genes. But you have to have enough memory to do it, so you don't want to do it on a laptop. And then there's some very excellent phylogenetic tree analysis tools. This is RaxML. Again, you don't run it on a laptop. It's extremely good. We have a very fast one, but it is not as exact. But this will also assign confidences to each of these branching points. You can then annotate these trees with any kind of information you want, like temperature, and so you can look up into other databases, where do those things grow, what temperature are they happy at, and then you have to bring in some scripting, because once you've decided there's a series of steps that you like to do and you want to repeat it, it's best to write it into a script there. And then we've expanded these tutorials on translation to the synthetases, and then to these other molecules that are involved in translation. So, okay. So, there's several there, so maybe when we get to it I can indicate to it. So the molecules that we'll look at here, as I said, are all the protein RNA complexes that are involved in translation. And so we look at evolutionary analysis, and also one's occasionally interested, is there an evolutionary a thread even to the dynamics that you see. So these are these proteins, I talked about the synthetases, that read the tRNA, they set the genetic code. So always the first couple letters are for the amino acid. It's supposed to be recognizing and charging up here. That's aspartic acid. It recognizes the tRNA for aspartic acid, and it has the codon in these positions here that the protein has to recognize. So it says, oh, okay, you're the tRNA for aspartic acid. I'm going to charge the amino acid up here at the CCA end, right? So you charge it. Now what happens to that charged amino acid? Well, as you know, it has to get to the ribosome for protein synthesis. It gets transported there by the elongation factors. I should say over here I've color-coded the structures by all the structures that were available for bacterial systems. So you can see there's a lot of conservation among them. Over here I color-coded the interface of the EFTU with the tRNA by the conservation in the bacterial sequences. So again, there's a lot of conservation in certain regions, and these space-filling residues here, these nucleotides, are modified bases, because if you're going to do RNA simulations, get used to the fact that there are not 4 bases, there are 110. So there are over 100 modifications of tRNA and RNA, ribosomal RNA, and they tend to be different for each of the domains of life. So if you even use E. coli to express a human gene, you probably are getting some of the modifications from E. coli in there. All right, end station for this is the ribosome. So if you notice, the tRNA is getting smaller and smaller, and now there are 3 of them sitting here on top of the messenger. I'm only showing you the large subunit here, this protein RNA complex, the large subunit of the the ribosome. So there are many, many papers that are on these. At the beginning, we were about the only one doing simulations on this first couple systems. So if you're interested in them, well, I should go back here. So that was one ribosome. And if you look at how many, if you want to follow translation inside even a small bacteria, this is a tomogram from Wolfgang Baumeister, and there are about 3,000 in this. This is Spiroplasma melliferum, and he's color-coded the large and the small subunits, blue and yellow. So if it takes you a long time to do one ribosome, this is an impossible simulation on an all-molecular level, an all-atom level. Okay, so again, the citations. This is the protein that sets the genetic code, and we've been looking at the structures since about 2003. Then he gets through various papers about, here was the one where we tried to find that missing synthetase for Carl. How does signaling take place? How do you read the codon region and tell it to charge at about 50 angstroms away? And then these are various modifications that can occur. And how does the ATP even get into the active site when you're trying to do the charging? For the EFTU, that's extremely interesting problem of recognition of a protein with RNA. And again, one of the things we were interested in is its interactions with ions. So in the cell, and when they crystallize these things too, they have a ton of magnesiums in there to be able to crystallize the structure. The RNA in general has more motions than the proteins do, but what's in the what the crystallographer uses is really unphysical. We did calculations with 2 different concentrations of the magnesiums, and I'll let it run, and you can see, compare the differences here. So over here, which is more physiological, this stem here, sort of the bases flip a bit and come back. Over here, the ions just go up and down the grooves, and it doesn't open. Now, this is still an ongoing hot topic. Are the parameters for magnesium correct? And a thing I find amusing, since we talked a little bit about the force fields yesterday, is everybody claims they parameterize them against the same NMR experiments. And they have totally different sets of parameters. So just how much motion, how much fluctuations you should be seeing in these, it changes because they just are still debating how best to be handling this. But it is in general much more flexible than when you're working with proteins. So a lot of these techniques that they were talking about to speed things up, you know, you do 2 femtosecond time steps, you better not do that with RNA, okay? And then as you move on to the host cell level, that's where we're going to talk about these papers that are down here at the very end. So how can I simulate ribosomes and ribosome assembly and translation in an entire cell? Okay, so some of the basic principles for evolutionary analysis is you should get used to comparing not only the sequences, but now there's enough structures out there you might and should be looking at comparing the structures. Realize that the alignments oftentimes need to be editing. As I said, I always like to go back and compare them to the phylogenetic tree based on the 16S rRNA, the reference tree here. And one of the things that you're going to get used to is not every protein is going to follow that tree. There has been an enormous number of cases of called horizontal or lateral gene transfer, where even the gene that we have in us is actually a bacterial in origin. Because I think it's no secret now through this human microbiome, we are populated with millions of bacterial cells. And sometimes we just give up and take over their gene, right? It's always good to look at your gene, where it is in the DNA. And if you want to try and get sort of a minimal profile of the evolutionary change of that algorithm, then try to use this QR algorithm to pull out representative members of that, right? All right, so what if you had a lot of multiple sequences? This is probably taken from the Swiss Prot database. I like it myself because it used to be, I think still is, one of the best curated databases that's out there. And this is, I think, like about, it's not all the 200, but if you have, say, 200 of those EFTU sequences, and you try to use what is called the classic clustral W alignment. That could take up to 5 minutes. This was back a couple years. There is an updated version. If you go to their clustral website, it's gotten much better. But there's like a competition even for multiple sequence alignments. Who can do the best? So one of the ones that we put in, and that was a request by one of the developers of MAFT. Was sitting in the audience just like you guys and said, could you make our stuff available? And if they allow us to export it with VMD, we do it. So there are other things called like tea coffee, also equally good. But MAFT said, sure, no problem. Just put it in and you can send it around. So if you do it with tea coffee, then the thing's done in 30 seconds, right? So that's really great. And it tends to be a little bit better alignment, I find myself, right? Okay. So one of the things we're starting to see already is the groupings. So we have a grouping there that says, oh, these were the alignments done with MAFT. You could also say, oh, these are the bacterial ones. So this notion of grouping is very important. How do you do the alignments? There are many, many, many programs to do a sequence alignment. The multiple sequence alignments are sort of at their core are based on pairwise alignments. And the most famous ones there are Smith-Waterman or Needleman-Wunsch algorithms. They're very simple algorithms. You have two sequences. You want to align them. And the algorithm, and you could imagine, there's an enormous number of possible alignments, the way that you could do these things. So they were able to break it down into a very simple algorithm that has the following steps in it. In the next step of the alignment, you can, so you're, say, at the one step before, you go and you calculate some similarity matrix. So this has sequences here, and how does arginine compare to cysteine, for example? And you've used some scoring metrics to set this up. So you look at the three possibilities. I either align the next two residues, and I look up what the score is, and I get a total score here, or I put in an insertion or deletion in one of the sequences. And in the simplest form, to put in an insertion or deletion, there's a certain penalty for that. In the simple case I'm showing you, I just made it -6 because it's sort of normalized to what these other guys are. So let me, if you really want to learn the details of it, there's a fantastic book written by Eddie on sequence analysis, probabilistic models of proteins and nucleic acids. And just to show you how this is done, here are two sequences. Here's a 4-letter and a 6-letter code. And you initialize it first with the values out of that similarity matrix. How does an M compare to an M with a G, so on and so forth. And then you do at the top initialization of the gap penalties. What if I put in 1? That'd be 6. What if I put in 2? It'd be -12, 18, 24, so on and so forth. Again, this is from an interesting website in Germany. Sort of gives you demonstrations of these very simple these simple algorithms. And now you start moving through using that Needleman-Wunsch algorithm. And so you can sort of see what they chose to do the first time was to align them. And even in the next step, it also chose to align it. This would be if you put in gaps here or to the gap there. So you can follow this all the way through till you get to the very end, and that's the final score for that alignment. And then you do what is a trace back. You follow all the alignments, and that's your alignment, end alignment that you get here. So you have M aligned to M, glycine to glycine. You put in a gap, and then now it's interesting. These two, if you had moved that over there, because of the simple gap penalty, would have had the same score. So that's the only problem with these multiple sequence alignments. There are often redundancies in it. And depending how the programmer has programmed it, it's either the first redundancy is the first one out, or the last redundant one is the first one out. So you might get slightly different alignments, and it just depends on how that algorithm was implemented. Now, if you go and try to align structures or do multiple structures, at the core is sort of the same thing whether you do multiple sequence or structures. you do a first pairwise alignments, get scores, and then you go through again and start readjusting everything. And there's some additional scores that go into this, like if you're doing structures. This is the one for stamp. They, Rob Russell and Barton developed this first for proteins. We then modified it to be able to do nucleic acids. And so there's an additional score that comes in there by looking at, say, the distance that you have between the two residues when you've aligned them, and also some local measurement of the conformation of that local segment of I plus and minus 1. All right, same thing though. You're going to try and improve those scores, and at the end of the day, you get this dendrogram of how it is. So first you do the pairwise, you see B was closer to D, A to C, and then you align them, and this is the overall alignment you get for all of them. Now, you have to always be a little careful with the stamp. If you have like that picture I was showing you in VMD, maybe it comes up again. If you try and align the whole structures, and this will allow you to do it even if they don't have the same number of residues, right? You might, it'll give you sort of a fuzzy picture if there's a lot of differences. You might, as you know your systems better, pick some local region that you really want to have aligned. You don't have to align over the whole proteins. It's like maybe the active site. Do that and then see how much variation is there. Or if you have a protein RNA complex, maybe you pick the proteins to align, or maybe you pick the RNA to align. So it's important that you use a little judgment on these things. So here's an example where we wanted to align two of the large subunits of a ribosome. One was from a bacteria and one was from an archaea. We wanted to see how similar they were. And they have two proteins out of I think it's about 30 on the large subunits, say L2 and L3, here and here. So I think we told it to align based on the proteins here. And here's the resulting alignment. The RNA alignments of the 23S and the 5S, that sort of goes around here or there, and the proteins are down here, right? But what's interesting is VMD, as far as I know, is still the only program out there that can align many, many, many whole ribosomes. And this is important because this is one of the darlings of the drug industry for antibiotic. This is where a lot of them go in and they try to attack the bacterial ribosomes or some residue that's in the channel where the protein synthesis takes place. So if you go into like Tom Steitz, who got the Nobel Prize or was one of the Nobelists for the ribosome, he has lots and lots of structures of the large subunit with various drugs in the tunnel, in the exit tunnel. All right, so now just a quick few words about evolutionary analysis. This was Carl Vo's original tree. And you can see with this tree that at this branching point, bacteria went this way, and the archaeas, which are single-cell organisms, are closer to us over on this branch. So there'll be a lot more similarity between these guys than between this guy and this guy, okay? For things involving translation. All right, so that's the reference tree. And let's go take a look at one of those synthetases. Let's look at the one that loads leucine. And again, this was taken out of his article back in 2000 that he did with Dieter Sohl at Yale and Michael Ibba at Ohio State. You can first see the 3 domains of life, but look at the numbers. There's so many more bacteria here. At that time, they only had a couple eukaryotic systems, and here were the archaeas in there. And as I said, the horizontal gene transfer has taken place. So sometimes the bacterial ones are the ones that you see in a eukaryotic system, or maybe these two have shared something. So think of that tree of life as having little connections or possible connections. So in describing sort of this variation, you often use the words full canonical, basal canonical, and non-canonical. And if it's full canonical, it just means you see these 3 sort of sections, with the archaeas being closer to the eukaryotics, and here's the bacteria. If you sort of lost one of them, like it's clear the eukaryotic ones have now, or sequence-wise, more comparable to the bacteria than they are to the archaea. And then sometimes you can hardly see any pattern whatsoever. There you have to be a little bit more systematic, and if you first look at just the bacterial ones, then you will see that most of the things in the eukaryotic system came from a couple sets of bacteria, right? So to quote one of the co-authors of that paper, so this horizontal gene transfer can erode the historical trace, but it doesn't completely erase it. But this strong pattern is you see really in things in the universal networks, like translation, to a certain degree also replication. All right, now to compare the structures in VMD you have a couple different metrics. You can certainly use RMSD, but then you have to have the same number of atoms, so you have to pick exactly the same number of points. Or as I said, there's this other algorithm that we use. It's called the QH measure, and so it gives you a score first for the aligned parts, and then how far apart are the gaps. And so here's a better example of this. So those two look like they're really aligned well, so that would give a score of about 0.82. Here this gap is growing, so the score keeps going down, and now you have this whole loop in there, so that'd be the lowest score. So if you took all the class 1 synthetases and compared them, and you just looked on the QH score, that's this blue one here, the light blue, and it's shoved more to lower values. If you just did a Q score based on the aligned parts, it'd be up here. So everybody would have a Q aligned score of about 0.5. These guys on average would be about 0.4, maybe a little lower, right? So again, it just takes getting used to this. One of the other points I want to make is the evolution is in both the sequences, so this is Carl's work, as well as the structures. So they had this set of sequences, and it was an interesting thing because if you look, it looks like, oh, you know, up here, there seems to be a bacteria up here, which should be down here, but you notice there's a 1 and a 2. So there was and this Deinococcus, that's a very interesting bacteria. One always says that if there is ever a nuclear war, this will be the guy that survives. You can scramble its genome and it keeps on functioning. And so when people were beginning to do structures, they really wanted to try and look at this synthetase that loaded aspartic acid, look at it in these two different organisms. What's the difference here? So the first thing you notice is there are always fewer structures than there are sequences. But here's the differences. So if we take everything in the archaea branch and align them, this is what you get. And again, it's colored by structural variation. So that part's really conserved, less conserved, sort of a new element in one of them. If you go over and look at the bacterial branch and align them, they look pretty similar, but it has this huge bacterial insertion that that guy does not have, right? Now, I won't bother you with this. Perhaps I'll go on to the next example. This is more like if you're looking at structural homology here and you're trying to think, I did it over the class 1 synthetases and the class 2 synthetases. They have a basic fold that's different in the core. As you get closer to higher structural homology, then just use sequences to compare the differences. If you get further back into those synthetases that are maybe of a different functionality, then go compare the structures. And I think I demonstrate this very nicely in this picture. So here are the class 1 synthetases for those ones that load lysine. Here is the class 2. They do the same thing. But look at the internal structure here. I think you can see these are parallel beta strands in the core. These are anti-parallel. So that's been one of the real problems with protein structure prediction. Even if you know the function, there can be two totally different proteins that do absolutely the same thing, have a totally different motif to them, right? And there's a lot of conservation of these class 2s across all the domains of life. And now let's take a look at just comparing some synthetases. Say this is the same synthetases, but I'll just look at two different X-ray structures. One that had maybe the AMP, the ATP in there that helps with the charging. Somewhat comparable resolutions. But if you look at the QH score for this, the structural score, it's 0.8. The sequence identity is 100%. Now let's go over here and take a look at two synthetases. They're from two different organisms. One's Yanashai and one's in this thing I can't pronounce. Right? And if you look at the QH score, it's actually higher than over here, and the sequence identity is only 70%. Right? So as I said, I put these more in just that you're aware of some of the difficulties that are out there when you're trying to compare sequences, compare structures. And if the structure is available, do try to use it, because Carl laughed. We spent a lot of time correcting his sequence alignments for those synthetases as we got more and more structures. They would be off whole chunks of the alignment. All right, so a final word to this. Like, let's say we looked at the evolution of, say, starting with a synthetase that would just, charge one of the amino acids. Alanine is always considered one of those first amino acids that came into being. And now you go through several cycles of evolution till you get to this point down here. We are now at a synthetase. It's in the same class, but it can now actually recognize aspartic acid. And if you look at sequence identity of those synthetases and their structural alignment, you see you're really down here in a range where these two sequences, this yellow dot here, they only have about 5% sequence identity, but structurally they're about, 45%. And here's the overlap, and you can really see, yes, they have exactly the same core. There have been a lot of additions in one of them over the other of these connecting groups here, right? But the core is absolutely the same, structurally. All right, quick word to getting these non-redundant representations. So we immediately went from having too little to too much information. So if you look at this, and so some of these synthetases, you can't even get it on a page anymore, the tree. There's so many. Because every time they sequence a new genome, they go and get these guys and write down all the synthetases, the elongation factors, and the ribosomes. So what do? I want to have some representative set of that. So we have an algorithm that encodes either the structure or the sequences in a matrix where you have all, say, 129 proteins this way, the length of the alignment this way, and then X, Y, Z coordinates and a coordinate for the gap here. And this algorithm, this QR algorithm, selects out of these the ones that are most different, right? Linearly independent. And you can do this both with structure or with sequences. The parameters have been set in the code. I think you can change them, but I wouldn't, because we had to go do this over a lot of different proteins to try and make it most useful. And so now if we looked at sort of the evolution of this synthetase that loads aspartic acid, let's look here where we're comparing things that are further away. This is where you also had the one for serine or proline. So again, the main conservation you see is that core of the class 2s. And then if you get down to this point, there's a little bit more has come in. All these guys have the same anticodon binding domain. It's this beta barrel type structure there. And then if we go a little bit further, as we zoom in to the aspartic acids, but now between the archaeal version and the bacterial version, this is what they have in common. But if you go down the bacterial branch, you should start seeing that insert coming in. And then you can go to the one that's just in, say, E. coli, right? And then color code that by sequence variation. All right. So as I said, oftentimes the structures are much more conserved than the sequences. They saw that in a plot, 5% sequence identity, but a Q score of about 0.45. And if you're trying to build structures, if something's missing, keep this in mind. It can help you build it more wisely, the missing residues. And so now I think you understand all these tools that we just mentioned here. So I still haven't answered your question yet about the clustering analysis, other than to tell you this clustering analysis is going back and looking at all the different cluster analysis that are in the alignment programs to do them. You do it pairwise, these dendrograms. Each one is a little bit different. Unless we developed the algorithm ourselves, we tended to use the clustering methods that were developed in the application of those algorithms. Okay, so now just to do a blow up so you can see it a little bit better. So now we have used these groupings. Here's the tRNA and their structures that are there. Here are all the sequences of the synthetases. And I think you can see very clearly the synthetases have a lot of sequence variation, but the structures are very much the same. And they have been color-coded here by the structural comparison. And if you tried to do it over the tRNA, you might get a slightly different alignment, or if you try to do it over the proteins, right? All right, so, and what's really good is when I was using this, doing more molecular problems, I would often map the sequence variation onto my one structure I'm looking at, just to say, oh, God, don't fall in love with that residue. It's a gazillion different things. You know, it's not such, cannot be that important. If you're thinking about in terms of the interactions, look instead where things seem to be a bit more conserved. Okay, just a word to the wise. If you want to load up all those databases on your desktop, that's completely doable. This was back in, I think, 2013, I think. No, I think, yeah, there are 1.2 million sequences now. And green genes went for about 100,000. This is up to over millions. So already to do this number of sequences for proteins or for RNA, this took about 2.5 minutes to load and took about 4 gigabytes of memory. This took 2.4 gigabytes of memory. You can put the whole databases in there. So, because it used to be that if you were trying to do something like BLAST, you could put in 1 or 2 sequences. When we were really working on translation and we wanted to look at all the 58 proteins involved in the ribosome, we were doing a large number, so we wanted the data right there because we weren't able to go over the internet to do this to the servers there. Now, as I said, one of the good features, particularly if you start looking at the circular DNA, if you're doing bacteria or the archaeas, it gets incredibly long. So you'd want to zoom down, color it by some feature that of interest, and maybe just zoom in on those particular areas that look interesting for you. The other thing I should tell you about with the trees, you can decorate them with a bunch of things. These are just the numbers, like the accession numbers, really unhelpful. It's far better to write in what domain of life they're from. And so they have here all the bacterial, material, that's what we call the metadata. And sometimes you'd even want to rearrange this. So you'll see there's some numbers at these branch points. That's giving you the confidence of that break, right? So this is pretty confident, that's pretty confident. This one down here looks less confident. So again, those are generated by the algorithm that you're using to do this. And if you're going to do the RAXML, which is very good, it has been superseded by yet another one, but you do have to do those offline, right? If you are doing something more than twice, I really strongly suggest you write some scripts. I think they talked a little bit about that in the first 2 days. It's also even like with NAMD, if you want to add in some other information, you can think about scripting it first. It's going to run a little bit more slowly. Like we wanted to add another force to something. Until it got written into NAMD, we just added it in through a script. Same thing, you can do this with VMD, and I think in the tutorials we have a few examples of those scripts. So one of the things, a new direction that is going, is you'll want to look at your a protein or your gene, where it is in the genome and what's nearby. Because sometimes, like particularly with archaeal and bacterial systems, there's this operonal structure. For the ribosomes, there are like about 8 or 9 main operons for the ribosomal proteins. And depending on what organism you're looking at, like E. coli, you can have 7 copies of the ribosomal operon, where you have the 16S, the 23S, and the 5S there. So first you can ask where they are, and this gives you the name. So this is like the 50S, the 30S, this is the polymerase. You can look for those. But what's really nice is first you construct a tree, and then you make the sequences align according to that tree. So you have the tree over here, right? This is comparing, say, the gamma protobacterias with the beta protobacterias. And now we're looking at some of this ribosomal protein operons here. So here's an easy one to view. This is Sec-Y. That's in the membrane. That helps transport membrane proteins into the membrane or into the periplasmic space. And we're going to zoom in on one of the proteins that actually was known to nucleate the folding of the ribosome, of the small subunit. That's called the S4. It's sitting in this one alpha operon in the majority of cases. So it's S4 here. But if you start looking at what happens to it, in some of the other organisms, it doesn't seem to be in the operon anymore. Well, where did it go? It moved into some other place in the genome, and sometimes it has 2 copies of it. So, and people looking at this found out some of these organisms go in when they are being challenged in starvation and make spores, and this is the region that generates the spore. That's the second copy there. Now, that's already a nice example of it took scripting in BMD to do it. This is such an important endeavor that there are now whole databases that are set up. So if you go to the ITEP, that does all the microbial genomes, and it has larger trees like this. So this is all the organisms that make methane that are archaeas. And over here, you can connect to them all their genomes, the entire genome here, but you do it by certain classification. These are known as Cod classifications. And actually what one was looking at here was not so much the conservation. One was doing this targeting in on, say, one of these organisms like acetophorans. And what we were interested in was the conservation of differentially expressed genes. So if you grew that organism in either a substrate with TMA or one with acetate, some genes were differentially expressed. So you looked at those, and then you wanted to see how many of them are conserved all across the entire genome, right? So again, this helps you sort of focus what makes it grow so much faster in TMA than in acetate. Let's zoom in on which of those genes is leading to that. So to me, it's not unexpected that it would be a lot in the ribosome because that's really going to affect the growth of the system, right? And then some of the other ones, the larger, are coming in and making the lipids. And again, things that deal with growth. Okay. What's another way to use a VMD, the multi-seq? So here we've taken the entire genome, the 16S, the rRNA, not the entire genome, the entire 16S rRNA. And we tried to do a study like Carl would have done. I compared the 16S among 2 bacteria and 1 archaea here. And you see these little red and blue stripes here. When you go back, you can look at the sequences. So Carl had a metric to differentiate a bacteria from an archaea. It had to have the same residue in all the bacteria, at least up to something like 95, 96%. And you can't make it 100% because there's still always errors in the databases. So those are the red ones. And then if you go and look at also where some of the structures changes are. These all had structures, so you could go in and take a look at them. You know, that's the blue. And if you look here at the number of bacterial signatures you have, and versus the ones that are in the archaeal here, you'll see this. This cluster down here represents all the bacterial signatures. This cluster represents all the archaeal signatures. And so one of the attempts now is to go in and find drugs that bind to the signatures that seem to be in most of the bacterial systems. So if you go in and look at some of the structures that Tom Stites has put into the PDB database, you're going to see a lot of them have the drugs that are binding to this residue, 2058. And when you develop bacterial resistance, or antibiotic resistance, what has typically happened is this thing gets methylated and the drugs no longer bind to that one. That was one of the most common causes of antibiotic resistance. So Carl had done it for the 16S, we did it for the 23S, and again you can see where the variations occur. And you have to be able to see this, you just use the zoom feature, right? So one of the main things that Carl asked for was this flexible grouping. So again, he wanted it like eukaryotic, archaea, and bacteria, because you know when he started off, they only had eukaryotics and prokaryotics. And he goes, well, look, these guys are really different than these guys. So he wanted to regroup them, right? So this is an absolute feature he wanted. But as I said, you can make this grouping anything you like, right? But if you bring in the information that's hidden in like this button, when you bring in it from the various databases, that nomenclature, that phylogenetic analysis will be available to you. So in fact, here it is. When you click on that button, depending on where you get the data, you get the sequence name. So again, this was the old notation for Swiss Prot. Tells you the organism it's from, the common name, it gives you the EC number. What's important is EC is not that unique. EC is more assigned to a function. So there are many proteins that can do the same function, but are really not unique. So like, again, think of that lysine, lysyl tRNA synthetases. Those are two different protein structures, two totally different sequences. So you'll always want to bring in the uniprot number as well. That is unique for every protein. And then Carl wanted a place to write notes to himself. Like, he showed me one of his notebooks, like never take another sequence from this guy's lab. You know, he got very upset about him. So you can keep that there too. All right, so that was a quick run through for the evolutionary analysis tools that are in VMD. As I said, many of these tools, particularly as you get towards comparing whole phylogenetic trees and you want to look at the entire genome, really go over, you can script it into VMD, but there's also a program, I think I wrote it at the bottom of this. It's called the ETE tree software. It's a Python based. So you should take a look at that. It's much easier. And you can talk to Joe Peterson, one of the TAs. This is a picture from a paper he's writing on the archaeas. Okay? So before we go on to applications of MD to protein RNA sequences and complexes, are there any questions on the bioinformatic part?
I have a very just general question.
Mm-hmm.
I'm just curious about it, Mike. I mean, how close are they to sequencing all the naturally occurring genetic sequences? I mean, you say there's like 1 million on this database, but I mean, there's certainly a finite number of sequences.
You know, we've got 3 different funding agencies doing this. And so like DOE cares the most about the microbial ones, particularly like the ones in the methane producing, because they make methane, right? And they're also used for cleanup of everything from oil spills to radioactivity. It's very selective, and you still have to write a grant to get the sequencing done. It's faster. So I would say as the speed drops, being able to do a whole genome sequence, pretty soon there's no reason why you just can't do it in your lab. Like there's this Oxford nanopore. It has a handheld device. You can sequence genomes very quickly. Of course, what costs money is the reagents that you have to put into it. So that's going to be your rate determining steps. But the big labs like JGI at DOE, you write a proposal to them. You have to justify why I want to have this many environmental sequences studied, right? Because it's just like us, no bacteria or archaea lives alone or is an island. They all exist, and there's a lot of subtle variations. So people want to, say, get ones, say, from fresh water, salt water, salt mines, nuclear waste, things like that. So it's very targeted, and you have to have justification because it's still not cheap enough to be able to do this. And of course, for health, they've gone through the major organisms. They've done the mouse, some of the major plants, the human, fly. But again, those are major initiatives, right? Okay, anything else?
So I guess I just wanted to comment again. I'm more familiar with proteins, but I mean, studying the sequences of data is also important in terms of understanding the structure of proteins.
Oh, heavens, yes.
I mean, so this is something that everyone should be interested in protein structure and function.
They should be. And, the thing is, there is a sequence initiative. That has pretty good funding. But where do you do the structures? Well, you have to have an accelerator, right? You go to Argonne, Brookhaven, out to Berkeley, and you have to get time on those machines to do that. So the structural genomics is, in some ways, behind. And at the moment, it's sort of funny. They actually have more of the structures have been done, but they don't know what they do. So but the Canadians are doing it, we're doing it, the Japanese are doing it, the Europeans are doing it. So they'll have the data there, but they haven't been able to assign function as quickly. So if you look up in any one genome, there's still a certain section of it is known, marked as unknown function. Maybe they've sequenced everything. They've got even maybe all the structures, but they can't assign the functionality to it. So if you stick with the model organisms like E. coli and yeast, I think yeast is one of the best studied. Well, and it's because of the wine and beer and bread companies. You want different flavors of beer. So they want to understand every little pathway, every gene, what it does in there. So thanks to them, it has the fewest unknown features in it. Right. Okay, anything else? All right, so then what I wanted to go through quickly is a few of the applications. So there are additional tools when you go into VMD. You can use multi-seq. You can also use it to compare sequence and structures. If you do NAMD, then you can bring back the energies that are involved in those interactions. So you can try putting it all together. Another thing that's really helpful, since you've already done the MD simulation on your system, you should do additional analysis of the data. And one of the easiest things to do is look at motions that are correlated in them. We developed our network analysis from an information perspective, but you can also relate those correlations to forces. And I think in tomorrow's session, you're going to hear about this elastic network theory. It's yet another approach to this. This is, as I said, is purely using your MD information, looking at the correlations, looking at the information, but other people have shown how those correlations can be tied to forces, and it's being used, this analysis tools, to study AFM experiments where you pull on things and you can, that really induces a lot of correlations, and you can pick up the paths and make predictions about if I change this residue, will the thing break faster or easier? And I'll show you one or two examples for that. Again, we were motivated from looking at these complexes, particularly the first one. How do you communicate the anticodon docking here with charging of the amino acid in that active site. So in that active site has to come an ATP as well as the amino acid, because the first step is to convert it to an adenylate and then attach that to the end of the tRNA. So let me just go through this quickly. So you would do your MD simulation. Now you're going to have, here's the synthetase, here's the tRNA. During that handoff, the EFTU will come up here and dock to it. There is no structure of all three of them together, but you can pretty much guess how this has to dock from looking at the crystal structures. People sort of knew this, but they tried to do it and they go, oh, it clashes, because they just tried to dock two X-ray structures. If you take the synthetase with the tRNA and do MD, you see that thing breeze like this. And it spends half of its time like this. And in that state, the EFTU can come in and start the transfer, right? All right, so if you're doing a typical MD simulation, well, one of the things you're going to have to decide on the force fields that you want to use. As I said, 36 is perhaps the best one in CHARM to use for RNA protein interactions. The amber ones were initially the leaders in the field because Coleman had dedicated it to looking at nucleic acids. Now they're very comparable, so just use whatever one that you're most comfortable in using. This 22 star was the one that D.E. Shaw developed, but it has not been tested out on RNA. He developed to do protein folding, so he had this very famous science paper where he folded, I think, 12 small proteins, and he found out to be able to have them fold into the crystal structure, you had to change one of the backbone torsions and two of the side chains. And so that later then was put into CHARM 22 star, but now, as you heard from Juan, it got a change in a lot of even the partial charges in 36, and the RNA protein stuff can be used quite nicely to study proteins. Okay, if you're going to publish, you have to be telling people all the parameters that you used, what ensemble, what temperatures, and if we are looking at this composite system, you'll have the synthetase, the tRNA, the EFTU, the GDP that is sitting in the EFTU, lots of waters. The composite system is at least 130,000. And if you want to see the tRNA move from the synthetase over to the tRNA and do it in a reasonable time, you might want to accelerate it by, it'll go through a bunch of forming and unforming salt bridges, and you can speed that up by just shifting into different rotamer states based on energy considerations. And Dunbrack has a very, very nice database. This is an old reference, but he keeps updating his website on this. So you can use his rotamer states to justify, okay, this salt bridge has been there for 10 nanoseconds. I think it's time for it to break, and then let's move on and go see what it picks up next in the transfer step. So you'll solvate, put in water, and run it. And as I said, now what to do with the simulations. So one of the things that we were interested in, here's the overall reaction. ATP and the amino acid has to come into the active site here and interact with the tRNA. So here's the protein, and it has to put this amino acid onto the tRNA, and the AMP and the phosphates have to leave. Okay, so again, here's where you recognize the anticodon, here's where you do the charging. So it's about 50 angstroms, and so how best to describe that signaling? Well, you can think of making a network, and you have to, network has nodes and edges to it, so where do you put the nodes? The example I'll give you, we just put them at the C alphas and at the phosphate of the backbone of the tRNA. You might want to shift to C betas or the midpoint of the side chains. With the nucleic acids, you really do want to do one in the backbone and one over in the base, so you can also pick up cases of differences in hydrogen bonding. Okay, so here are the nodes, and now you run your simulations, and you look at how many of those nodes are within a certain contact distance, which you specify, over the majority of your simulation. So the contact distance you have to play a little bit with. We suggest one that seems to be working for protein RNA. And when we say nodes are in contact, so if you put it at the C alpha, we mean any heavy atom in that residue, does it make contact with any other residue, the atoms in any other residue, whether it be protein or RNA. So it's just not C alpha to C alpha distances. It's any heavy atom in that residue interacting with any other heavy atom in the other residues. So draw a line. And now you could think about how's the information transferred. So let's say maybe this was the anticodon, one of the atoms of the anticodon. Maybe this is the active site. There seem to be 10 steps to get there, right? And if you look at other paths that you could take here, they will be longer. This seems to be the shortest path. This one was 10. But what you do now is you weight those paths, those edges, by correlations. So you have to go in and do a correlation map here. So you put the sequences here and the sequences here. And you calculate. In this case, we're doing the Pearson correlation. So first, this is just the nodes and the edges without any weights from correlations. And then here is the correlations. So you look at how much the position of that ith residue or node changes to the average, and then you normalize by the motions between I and J here. So you look at the covariance here. So these are the fluctuations of I and fluctuations of J. Are they correlated? And divide it and normalize it. And the weights are equal to minus the logarithm of this, because these correlations tend to go from like -1 to 1, right? So take a logarithm, it's going to make them all negative, so I want positive weights. So we put a negative sign here, and we put absolute values, because when we looked at this from an information perspective, just a second, we didn't care if they went this way or were correlated or anti-correlated. We just wanted to know that the motions were correlated. So absolute value, and then you could come up with this picture, so you can already start seeing some clustering by weighting the different paths. And now you can start asking, well, how many steps does it take me, or what's the weight to go, say, from here up to here, right? And add in all those weights for all those different paths. Now, you had a question in the back? Yeah, that's a really good question. So again, it depends. Like when you're looking at an equilibrium simulations, it doesn't hurt to do it in a couple snapshots. You don't want to take the ones right at the beginning, let it accumulate, and then if you're doing a couple hundred nanoseconds, maybe compare the ones at 100, do it for 20 nanoseconds, and then maybe do ones a little bit later. Like to simulate the reaction in the synthetases, what happens after it gets charged, the local environment changes, and the tRNA starts coming off automatically. Then you have the EFTU come up. So there, if you want to watch it being transferred, you're going to want to take 20 nanosecond windows during the whole process, right? And see it change. Because to be frank, loss of correlation is the biggest indicator that the two objects don't interact any further. And what's interesting, that takes place within a half of an angstrom. You know, it's quite, and you look at it, you almost can't see it, but you can calculate it and see immediately the loss of correlation. All right, so that's it. So let me give you a quick demonstration. Let's go from one of those residues in the anticodon domain. It's interacting with, I think this is like residue 34. So the anticodons and tRNA are always in certain positions. And so here are many of the paths leading up here. I haven't drawn all of them. I've drawn the minimal distance, the shortest, the optimal distance, and then the ones that are within a couple epsilon of that answer, right? So there is the shortest distance. But now if you take a look, some of the others pretty close. So if I maybe knocked out something in here, I might be able to take this other path. So people are trying to do those kinds of experiments. In fact, they do a better job at it than we do. There was a group, I think it's Loria, he's an NMR guy at Yale, did this. He actually went and changed the pH. So then he would change the pathway and he could recalculate how the correlation should shift. So very, very powerful use of this. And let me now rotate it around so you can see. So I've colored in red the optimal path, and you can see these pathways. Now, way we used it, again, we used it from an information perspective. A lot of network analysis that has been developed has not come from scientists, it's come from the social media. So people are constantly asking, you know, what group are you in? Are you a Boy Scout and a conservative? And do you live in Illinois? So the algorithms that we use were developed and published in PNAS, but they were used for really studying connectivity between different groups, right? So this is a particularly simple demonstration. You have 3 communities here, and there seem to be sort of major links between them. Here it has 2, this one has 1. So if you were trying to find the communities, doing it complete from a math perspective, you go along breaking all the edges and see when do you fall into, say if I broke this one, then suddenly I would have 1 community and then there's 2 communities. I'd have separate communities. So if I'm trying to find those communities, I go along and I break bonds and I see when this thing falls into separate entities. And these communities are, are defined, if you look, there's more correlation within than between. So here's a very simple case. I only have 1 connection. Here I got tons of connections. I can break bonds all I want here, and I'm not going to break that thing into a lot of different communities. So, so there, as I said, are subnetworks with many intra-community edges, but few inter-community edges. And the algorithm that we use is exact. There's no variable parameter, and it's called the Gurman-Newman. They're 2 different people, but one is a social scientist. And one of them, oh, I don't know if it's Gurman or Newman. One is a woman. She's now getting more into biophysics, so you might see more and more of her work on this. It's pretty good, but if you start getting to these huge complexes, then you might want to go over and use one that the French have developed to look at your cell phone usages. And it's not exact, so you have to play with it a bit, see what parameters will give you the best agreement on a smaller system with the Gurman-Newman. But this has, as I said, no adjustable parameters. This is what we use for these smaller protein RNA complexes. So I want to just end here with giving you one nice example. So now we're looking into the active site of that synthetases. You have the CCA end of the tRNA coming in. You've had already the amino acid has been charged, and it's now an adenylate. And now I've got to get that amino acid connected here onto that OH of the CCA here. And then the AMP should be leaving. So after the attachment takes place, which I have here, then here's the AMP, has to be leaving, right? So this was also a system that was studied by Stites before he turned his attention to the synthetases. So what we were interested in, if you look at this as a chemical reaction, sort of what groups interact with this part and what residues interact with this. And to our astonishment, when we did the simulations, and we just zoomed in on the active center, we saw 2 communities coming up, the purple one and the orange one. And that adenylate is sitting in here. You can sort of see the ring from the AMP here. And let's take away 1 so you can see it more clearly. So there's 1 community that seems to be interacting with the amino acid. That's the purple one. And there's another community interacting with the AMP. So if you were to try to do, say, a quantum mechanical calculation or a QMMM calculation, you might want to take at least some portion of these residues into the quantum mechanical part of treatment. And the same way with the purple, a certain part of it closest to the amino acid residue. It's almost like the networks have been set up to help with the separation process. So there you go. So as I said, we found this extremely interesting, how the interactions within the proteins have been developed to help motivate that cleavage reaction. And if you want to just take any of your simulations, this is with the protein, the synthetase, and the tRNA, I think when you analyze them, without charging, you'll see you'll have about 8 or 9 communities, and you can go through, and this is just a very simple clustering diagram that you can take out of the information that comes from the Gerwin-Newman. As he defines these communities, you take it and just put it in a simple dendrogram. And you can see the first 2 communities that are formed is the top part and the bottom part. So this is the part that is actually conserved among all the synthetases, and the bottom part is the stuff that has the modifications for the domain. And you can go through each one of those and see sort of what sections are moving as more or less one correlated unit. Now, all these techniques, as I said, we did not invent them, even the language. They all come from network theory. So one of the things we put in, I guess it's somewhat new, is to look at the optimal paths connecting certain residues up to that active site. An idea that Ruth Nuzinoff liked was she was really interested in those critical conserved nodes that connect the 2 communities, like those simple ones that we showed you in that very this picture with the 3 communities. And then finally, this is one that they use more when they're doing traffic analysis. So like if you're trying to study traffic control in, say, Paris, and you took any 2 points within the Paris map and drew the optimal paths between them, and then you look how many of those optimal paths go along the same streets. That's what you would be seeing. So you have some threshold. So all of those things that are in blue here share a lot of they share having a high number of optimal paths going through them. So typically with Paris, this would be like the Boulevard Périphérique, for example, would come popping up in these. All right, and then finally, I think you were asking about how often one should do this analysis. So here we have the synthetase with the tRNA. You can even look in this purple community. It involves the protein and the RNA. It's just not a protein community or an RNA community. And now if you bring up the synthetase and dock it reasonably without clashes and just wait, you'll see the tRNA, you've charged it, and it'll start moving off, and you lose this connection here, and now this community is entirely within the tRNA, and now you've developed a new one between this part of the tRNA and the synthetase. Right? And we did a nice demonstration of that in a FEBS article in 2010. And so you can really see the shift in these. And it just took about 20 nanoseconds there and another 20 nanoseconds over there. Okay, and finally an application of these correlations. So if you ever looked at this book by Callan on statistical mechanics, he has in chapter 19, equation 14, shown that the correlations of the fluctuations can be related to the force that's acting on these atoms. And so people who do AFM experiments, so here is a complex that's, it's a Dockerin cohesin interactions that are in cellulosomes. It helps the bacteria stick to the plants that it's trying to chew up, and you start pulling on those and do a network analysis. That's the optimal path, and that's where you're pulling, and that's where you're pulling. Right? So it's a beautiful demonstration of just how you can use these correlations. Okay, so finally, if you're interested in any of these parts, either the network or looking at evolutionary translation of any of these molecules involved in the translational pathway, we have tutorials. They're online. If you want some of the data to run them and don't want to download the entire databases, we have a small section of them where we've just pulled out the interesting molecules, and we have them in a Dropbox. I have to ask Joe if you guys could get to it. Put an end to that. Okay. If you're interested in doing those. All right, so we're done a little early. One thing I should do is mention who developed these tutorials. So some of this work was part of the thesis of Kay Chen, Jonathan Lay, John Ergel, and Elijah Roberts. There's Elijah and there's John, and Elijah's a professor at Johns Hopkins, and Ka is out with Bernard Paulson. She's shifted to doing metabolic network analysis as we move to the host cell, and she's in California, and he's just finishing up. And now we have 2 new TAs that have come, but they know very little bit about the molecular world. They are our experts on the cell. So if you have questions, I'll try to help you. And these are our programmers back in Urbana. There's John Stone, who develops VMD, and Kirby, who is just now gone into, I guess, real estate, just left the resource, but he was helping also implementing a lot of the, and updating the tools into the bioinformatics. So, as I said, you're entirely welcome to do them, but we're here really to have you see our latest version of the cell simulations. And to do them, we want to make use of the Amazon cloud. So we have a node for each one of you, a GPU that we've reserved up on the Amazon cloud. So in the afternoon when you start, you should be able to go through the exercises very quickly. And then you can visualize the cell simulations in VMD, right? So we'll see how this works, because we didn't like the connection to, we used to get you guys all accounts on Blue Waters for a couple days. And the trouble is the time change. Here it works for you. When we got to Berkeley, we suddenly forgot 5 a.m. in Urbana is 3 a.m. in Berkeley, and they shut off the account at 3 a.m. in the middle. So, no, we're not doing this again, right? So we're going to try out the cloud. It works pretty well. They've been testing it. The only thing that I can see is that we have much faster GPUs at home. So we truncated the exercises a bit, had you run them not quite so long, so you can get them all done. I think the longest one takes, is it 15 minutes, Joe?
Yeah, it's about 20 minutes.
Okay, so we'll put that one to the end. It's on looking at the min oscillating reactions that just precede having cell division, so that you can see that. Okay? So right now, any questions on the molecular level? And we'll go for a break. Yeah?
I have a question about the setup of the RNA simulations. You showed the
Sure.
time step of 1 femtosecond.
Yeah.
Well, generally, when you put rigid bonds, you use a 2 femtosecond time step. So I
You
was
don't
just
put
curious.
rigid bonds.
Oh, okay. So we just don't put rigid bonds.
Yeah.
Okay.
And even if you did, I shouldn't say that. I think sometimes they do. The reason you don't do that is, RNA is an electrostatic monster. And so if you looked at that Verlet algorithm, that last term, it was like t squared, and you had a force. Go to the next one. The errors just get to be too large. You have the electrostatic forces are so strong, you don't want to take large time steps.
Thank you.
Okay. Anything else?
Have you looked at the direction of information transfer when you have correlated functions?
Actually, it shows you the information pathways, because we've taken the absolute values of the correlation. This motion and this motion, correlated and anti-correlated are the same. So it only tells you that it shows you the pathway, and it's correlated. And as I said, we did that from an information perspective. If you go over and then try to look at the the correlations and those fluctuations, there you can relate it to forces. But again, the force in I to J is the same as J to I. You just change the sign. Okay? Yeah.
Okay. But the thing is, I was wondering, like, if we can identify other drivers and responders, right? Because if we have two communities, right, one has to be dominant and one has to be respondent, right? And it has to be.
Okay, so typically, like when you do the AFM examples, you fix one end, like with a peg, to a surface, and you have a cantilever, and you're pulling in one direction. So you say you pull on the C terminus of one. So I think that's a good question. And you just pull till the thing snaps apart. So you're wondering, well, in all the cases that we use, that we've developed, we just look at the optimal paths. So it's always, I to J, and as you go along the path. I think you're saying is like, well, if one community has a lot of connections, is that going to be more the dominant one?
Yeah, like, if you can identify those.
Yeah, but to me, there'll be a lot of communication within, purely from an information perspective. I haven't thought about it from an energetic perspective as much. Those guys like Hermann Galb and my husband, they're looking at it more from an energetic perspective. But what's essential are those critical links between the communities. So if one has only one and one has two or three, then obviously the one with two and three is going to have a tighter connection, right? But to correlate it with the number, I will say that we tried to do, we have used the correlations to get at relative binding rate constants or equilibrium constants. And I would love it, I tried only very briefly to do a statistical mechanical theory about if I looked at the number of paths within a certain epsilon of the main optimal path, then I did a mutation and I counted those paths again, can I predict what the delta G's? It worked in one case, but one case is not, is no case, to be frank. It is just, I could have been really lucky, right? Okay, anything else?
What networks you had the cutoff 75%?
Yeah, for the majority of the time, because if you put anything 100%, then you had to make the window too small, the window of time. So we felt it was a good compromise. In that paper, do I have it up there? Yeah. We first used this, oh boy, dynamic signaling networks. That was in a PNAS paper in 2009. The SI has a table of, we tried different times, we tried different threshold distances. It depends on how you set up the physical network. So one of my former students, Romy Amaro out in California, they looked at moving the nodes to the average position of the side chain. They felt that was even a better predictive value. Well, then you might want to, I don't remember she adjusted those thresholding times, and it's just, you do it on a few systems where have a good idea at the answer, and then you continue to use those. So in that sense, there is an empirical part of applying these things. I just can't give you here cookbook. It depends a bit on the architecture. Okay, anything else? Okie doke. And then why don't we go ahead and take the break? It's a little early. And Mike and Joe, is there anything I guess we wait till this afternoon and we would give them the accounts. Is that correct? So you started them all? Okay, so then you'll get them after all. So after the break, then we'll talk a little bit about the cell simulations, because it's a totally different approach. It's no longer all atom. So we'll have to explain a little bit of the details, what's going on. Mm-hmm.
That would be good for, like, when you do the full cell. That would be good for understanding pathogenesis of diseases, right?
Yes, it can be. One of the things that we're interested in, if you stick to the model organisms, like with yeast or with E. coli, you can get really predictive. And I'll show you a great example of where we could predict within a colony of cells how their access to the resources change their metabolic behavior, how cells at the bottom near the food behave differently than the ones at the top and the ones in the middle. So it turns out this thing that you're asking about, it's called metabolic reprogramming, and it goes on in cancer as well. But then it's access to blood vessels, right? And we share acetate, and in human cells they share lactate. Yes, we know. We showed that first results, and we had every cancer person in the room come up and tell us, why don't you apply this to this? So we're trying, but try getting a good model of a human cell. So first we had to do yeast, because that's a model eukaryotic system, and we feel if we understand that, then we can modify it. It is at the basis of all the reconstructions that they do to develop the networks for the human cell. So we've been working for 2 years to get a really good model for it. Right, so yeah, you're totally right. I'll put those slides back in. I didn't know you'd be interested. Uh-huh, uh-huh. Right. Oh, yeah, okay. So we're actually working on a model of the smallest pathogen with the Greg Venter Institute. So we'll have a full all-atom as well as a cell model for that, a coarse grain model. But that'll take us about 2 years. We have the coarse grain first step sort of done, but it's going to take a while, because that's like getting into 2 billion atom simulations. And we have to change a few things in MD and BMD. Okay,